I'm Owen Peters and I'm a lecturer in the UK DOI at Cardiff University. So I'm trying to understand the molecular mechanisms that underlie neurodegeneration using a range of model organisms and cell culture systems. At the moment, we're aware that Alzheimer's disease is a very complicated disorder. The genetics of it aren't easy to understand. It's not a disease where a single gene causes the disorder. What we're beginning to understand is that actually a large range of genes interact with one another to cause the disease. So the challenges at the moment are firstly trying to identify these very rare genes and then to understand how they change how cells function. So the brain is made up of lots of different cell types, and one of the first challenges really is to understand which cells these genes even function within. We're starting to find that these genes now function predominantly in microglia, which are a class of cell that aren't as well studied in neuroscience as neurons are themselves. So Alzheimer's is genetically a very complex disorder. So we know that it's not single genes that cause this disease in the vast majority of cases. We're starting to understand as genetic techniques become more powerful that Alzheimer's is most likely caused by combinations of very rare risk genes. So the challenges at the moment are first trying to identify these rare genes and rare variances, and then also trying to understand which cell types these genes are expressed in. So historically, people have focused a lot of Alzheimer's research on looking at neurons, but now as genetics is starting to unravel some of the underlying factors that cause this disease, we're starting to realize that microglia are actually probably a very important cell type, and these have been historically rather understudied in Alzheimer's disease. So the challenges are going to be overcome through funding, increasing expertise within the field, and through international collaborations. So one of the big challenges with genetic studies is trying to actually get enough samples together to be able to run very powerful statistical analysis. So this really requires us to have these very large experiments where you have tens of thousands of patients to hundreds of thousands of patients. So I think over the years now, international collaborations have been growing, and I think as that continues, that's going to be really helpful for the field. So at the moment, we know a lot of genes, but we don't know what a lot of them actually do. So one of the challenges is going to be really trying to use the correct analytical techniques to try and understand these genes. So for example, from the work my lab does, we try to use model organisms to try and understand how these genes function within the intact brain of an aging animal. So we do that using fruit flies, but these experiments can also be done in a wide range of model organisms. There's also current techniques to look at cells derived from patients. So techniques such as induced pluripotent stem cells allows us to make neurons and glia from skin cells from these patients, and then actually try to look in these tissue culture systems how these genes contribute to biology and degeneration of these cells. I hope the field will grow. So I think it's very ambitious to hope that we would have any cure within 5 years. But I think at the moment, the UK DOI really has brought in substantial funding that has allowed the field to grow within the UK. And I think that's really what we should hope for over the next 5 years to the next decade, is to expand the field. It's very much like how the cancer research field was 20 years ago. Investment and training is going to be really key to the field.